In the last lecture, we talked about different types of biotechnology and the history of biotechnology. And in this mini lecture, we're going to talk about sort of the tools and techniques that we use in modern biotechnology fields. I am not going to go wildly in depth on these. And the reason for that is, honestly, if you're not a biologist, you're never going to touch these again. So I'm going to give you an overview of the tools, an overview of the techniques, but I'm not going to expect you to be able to immediately use these tools and techniques until, your second year in college. We can use this recombinant DNA to move fragments of DNA around into new arrangements. That's how we got the glowfish, the ones you can just go to the store and buy here in Virginia. There are a lot of states, by the way, where it's illegal to buy glowfish. Yeah. Because they're transgenic. That's just because you're terrible at raising fish. We have two types of techniques we can use, in vivo and in vitro. And there's a difference. And it's a really easy question to say, what's the difference between in vivo technology and in vitro technology? Right, in vivo is you're injecting it into a living organism. In vitro is where you alter DNA outside of the organism. So in vivo is directly getting the DNA into the organism, and in vitro is you deal with it outside and then you inject it in.
So for the people that are getting in vitro insemination, right?
They create the organism outside the body.
And then they put it in her.
Correct.
In vivo.
Right. Well, it would be in vivo if the procedure was done inside the body. But it wasn't created inside of her. It was created outside.
So in vitro is just planting the fully fertilized zygote.
Right. Now, back in the 1970s, this is where all this biotech came about. You're at Stanford University, and they took these two isolated DNA fragments and bound them together. So you took DNA and stuck them together, and nobody knew that could happen until the 70s. It was huge, because once we figured out how to connect two pieces of DNA, and it still transcribes, it still replicates, now we can start changing the structure, the fundamental structure of our instructions. So after they built this DNA, they stuck it into a bacteria, and that bacteria started making whatever that gene product was. So they created a new polypeptide. This is the real structural foundation of transgenics. And I tell you that because this helps us understand, this ultimately for all of biology, it was a huge leap forward. We don't have to just observe what's going on. Now we can directly experiment with genes. We can turn things off, we can turn things on, we could figure out functions. And that's what led us to be able to interpret what all of our body, what our instruction manual says. That led to the field of molecular biology, where we're routinely using cloning and genetic transfer technique in order to figure out how the world works. Gene cloning is where you're going to make lots, and go figure, right? You're going to clone something. You're making lots of identical copies. The first step is a restriction digest. This restriction digest involves enzymes that cut DNA at specific pair sequences, and that creates many, many fragments of sticky ends. Now, this used to be used, or this is used in cells in order to defend the cell. The cell will seek out specific viral sequences and start cutting them up. We've taken this for ourselves to make it attack very, very specific regions. Once you've cut the DNA up into little pieces, you can use gel electrophoresis in order to purify that piece. So DNA itself is negatively charged. When you place it in an agar gel, or sort of like a jello type substance, you run an electric current through it, and the DNA is going to travel toward the positive pole. Negative is attracted to positive. As it's moving, it gets separated out by weight, because the heaviest stays farther behind. It doesn't move as quickly. It's harder to move. It's like, I don't know, if you were to move a wheelbarrow full of rocks compared to an empty wheelbarrow. The one that's lightest is going to move fastest. So you get these different bands. Next, you take those bands, you purify them so that you have the extracted DNA of the specific region you're looking for, and you run a polymerase chain reaction on it. Here, the DNA is going to get heated up to break down the hydrogen bonds holding it together. You denature the DNA. That's then followed by a high-temperature DNA polymerase binding to the single strands. That's called annealing. This high-temperature polymerase came from a thermophilic bacteria, thermophilic agrobacteria. It lives in hot springs. It's called Taq polymerase. You don't really need to know that, but it's interesting. So you break apart the DNA. High temp DNA polymerase binds to it. Then you lower the DNA. You lower the temperature, and the DNA then begins to bind back together. So denaturation, you heat it up, you break the hydrogen bonds, you add a high temperature DNA polymerase that creates a copy of the DNA, a single-stranded copy, and then you lower the temperature, allowing those copies to come back together. You repeat this over and over and over again, so you can go from a single strand of DNA to millions upon millions of strands in a very short amount of time. Next, you could take that piece of DNA you have and insert it into a genome. Often you need to use ligation for that. DNA ligase can take those sticky ends of DNA and connect it to a new strand. So remember, we use the restriction enzyme and we cut, creating sticky ends. Here's where you put those sticky ends back together and you reconnect DNA to another strand of DNA. So we can now recombine DNA. We can take a bit of DNA from one organism and a bit of DNA from another, or a piece of DNA and then add a wholly from scratch designed piece of DNA to it. That's called genetic engineering. You're creating novel or new DNA sequences. Typically, it means cutting DNA at very specific points using restriction enzymes and then gluing them back together using an enzyme called DNA ligase. One of the most important techniques that's used in recombinant DNA technology is PCR, or polymerase chain reactions, where you're going to make many, many, many copies of a very specific gene sequence. It amplifies it thousands upon thousands of times very quickly. This is something that you'd start and run overnight, or just for a few hours. Start in the morning and then it runs until the afternoon. PCR involves a series of temperature cycles. So you denature the DNA, you split it open, then you cool it down so a polymerase can attach. That DNA sequence will extend, and then you heat it back up, and you repeat the process. You get a lot of cloned DNA very, very quickly using PCR, so that you don't have to worry about growing it, say, in a bacteria. It makes the processes of recombining DNA exponentially faster than they were, say, 15 years ago. So in this mini lecture, we talked about a bunch of stuff. These content review questions are here to focus your studies. We talked about in vitro and evo technology, restriction digest, electrophoresis, and so on. And then I really focused on PCR. In the next mini lecture, we're going to talk about how we can use these technologies to enhance plant products. We'll talk specifically about plant bioengineering. So that's the next in this sequence, is plant biotechnology.